Hello everyone. Today we will do quick revision of the chapter Biotechnology Principles and Process. This chapter is very important for your board examination for class 12. Now first we have to understand what is biotechnology. Biotechnology, we know basically it is combination of two words. Biotechnology, it is made up of two words, that is biology and plus technology. When biology it is combined with technology, biotechnology is formed. So biology, we know what is biology. Biology is basically science which deals with living organism. So science of living organism, it is known as biology. And when this biology it is combined with technology, it is known as biotechnology. So when these living organisms are used for various purposes, for the mankind, for various things, and we are using different technologies for the improvement of like we are using these living organisms for making vaccines, for improving the environment, and we are using different techniques also. So this is forming the biotechnology. So earlier we know that biology was used, but it was not very effective because we were using only biology, which is the science of living organism. But nowadays this recent branch is there which is known as biotechnology. So why this biotechnology is there? Because initially biology was not very effective, because biology is only the study of living organism. And so earlier when there were diseases, we used to there were many tests, and but there were no past techniques were there. But now we are combining various techniques with biology, and these techniques are very effective to find out the various disease. Various markers are there, and there are various new markers, and with the help of these new markers we can find various disease. So this biotechnology, what is this biotechnology? Biotechnology, it refers to technology using biology, and in this biotechnology it has various applications. Like it can be used in agriculture, it can be used in food processing industry, it can be used in medical diagnostic. I have already told you that this biotechnology, it is very important for medical field. Like we can find out various diagnostic markers with the help of this biotechnology, and it can be used in medicine. We can make various vaccines. Like in the recent past we have made, which vaccine we have made? We have made corona vaccine using this biotechnology. So it is very effective, and this biotechnology is also used in bioremediation. Further, we are using this biotechnology in waste treatment also. How we can use this biotechnology in waste treatment? Like we can recycle the waste. We can recycle this waste, right? We can make this waste useful by recycling this waste, and energy production also. So this biotechnology is very important as it is having multiple applications. So as compared to biology, it is very useful. So this EFB, that is European Federation of Biotechnology, it defines biotechnology as integration of natural science and organisms, cell parts and molecular analogs for products and services. So what is biotechnology? Basically biotechnology, it is application of technology to industry, agriculture and environment. We can use this biotechnology in various fields. Like we can use this biotechnology in agriculture also. We can use biotechnology in agriculture. We can use this biotechnology in medicine. We can use this biotechnology for waste treatment. There are various applications of biotechnology. So this is the reason nowadays many of the students, they are opting for biotechnology. Earlier students used to take biology. Earlier there was no biotechnology, but nowadays we have combined biology with technologies. So definitely this is having much more scope as compared to biology, because we are combining this biology, study of living organism, with various techniques. So definitely when we are combining this biology with various techniques, it is much more effective, because we are able to find out various markers. We can find out various markers, we can find out various disease. Earlier there were labs, but there were no technologies. So we used to take it was when we have to diagnose a particular disease, it used to take 3 to 4 days. But nowadays there are different techniques. So with the help of these techniques, we can definitely diagnose a particular disease in less time. So definitely this modern, there are two basically techniques of modern biotechnology. What are these techniques? You should know. These are the two techniques of biotechnology. One is genetic engineering. This is genetic engineering. So basically this biotechnology, it is divided into two techniques. One is this genetic engineering and second is this bioprocess engineering. Now you have to understand what is this genetic engineering. Genetic. What do you mean by the term genetic? Genetic means, definitely it is the term which is concerned with the genes. Somewhat it is related with the genes. And genes, you know, genes are basically nothing but they are segment of DNA. So what are genes? Genes are basically the segment of DNA, or they are the pieces of DNA which are responsible of the characters which are they are carrying the hereditary characters. They are carrying such hereditary characters which are transferred from parents to their offspring. There are certain characters which are present in your parents, and those characters are transferred to you, to the child also. So these characters are hereditary characters, and definitely these characters are transferred with these genes. So definitely, what do you mean by this genetic engineering? Genetic engineering, this word means, genetic engineering means when we are dealing with the genetic makeup of an organism. So what is this genetic? Genetic means what is the genetic material? You should know. The genetic material which is present inside an organism, the basic unit of this living organism. What is the basic unit of living organism? You must be knowing what is the basic unit of living organism. Basic unit of living organism is cell. So inside the cell, genetic material, basically there are two types of genetic material. One is DNA and other is RNA. So in human beings, this DNA is the genetic material which is responsible for transmission of hereditary characters. So genetic engineering basically means when we are engineering the genetic material. That means we are manipulating or we are engineering the DNA. We are making some changes in the DNA. That means whenever we are manipulating, whenever we are engineering the DNA sequencing or sequences, that is known as genetic engineering. So introduction of foreign genetic material into the host genome. Host genome means there is one person, okay? That person is having his own DNA. Now there is some problem with the host DNA. Now we want to introduce foreign DNA. We want to introduce some foreign DNA into the host DNA. So what we can do, we can bring that foreign DNA, and what we can do, we can insert that foreign DNA, like we can name that foreign DNA like A. A is the foreign DNA and B is the host DNA. We have to insert this A into the B. So what we can do, we can insert this this A into the B. So this is basically the genetic engineering. So if we are making some changes in the host DNA, host genome, when we are inserting this A, that is foreign DNA, into B, that is host DNA, this is known as genetic engineering. When we are making the changes in the genetic makeup, that is when we are engineering the DNA, host genome. So this is basically known as genetic engineering. So this is basically one branch of biotechnology. Now what is another branch? So what is this genetic engineering? It is basically introduction of foreign genetic material. I have already told you what is foreign genetic material. It can be DNA, it can be RNA, and what we are doing, we are introducing this foreign DNA, foreign genetic material into the host genome and altering its phenotype. What is this altering its phenotype? Now here you have to understand basically two terms are there. Basically two terms are there. One is phenotype and one is genotype. These are basically two terms. You should know the meaning of these two terms. What do you mean by phenotype and what is genotype? Genotype, basically it is genetic makeup of an organism. All the genes which are present in an organism. That means if an organism is having a number of genes, organism is having millions of genes. So all the genes which are present in an organism, that is known as genotype. Now you know that one particular gene is responsible for one particular character. That means if millions of genes are present in an organism, definitely they are showing millions of characters. So that character is known as phenotype, because we cannot see the gene, we can only see the characters, because genotype cannot be seen. Genotype is there in the organism, but we cannot see the genotype, we can only see the phenotype. So what is phenotype? Phenotype is basically the external appearance, which is because of the genotype. Okay, so phenotype is the external appearance, external characters which are present because of this genotype. So these are the two terms. So when we are altering this DNA and RNA, when we are making the changes in the genetic material, that is DNA and RNA, so what is changing? Definitely when we are making the changes in the genetic material, that means genotype is changing, and I have already told you that one gene is responsible for one particular character. One gene is responsible for one particular character. Like if you are having blue color of eyes, that means you are having one gene. One gene in your body is responsible for your blue color of your eyes. So if we are making changes in that gene, if we are isolating that gene from your body, which is responsible for your blue color of eyes, definitely your color of your eyes will change. How it will change? We can isolate that gene from your body, and what we can do, we can change, we can make change in that gene. How we can make change? We can make changes in that DNA by genetic engineering. We can alter the genetic we can alter the DNA. Like we can remove a particular sequence from your DNA, and what will happen? After some time, you will see that the blue color of your eyes changes to black color. Now, what had happened? You can see that because we are changing the particular gene. What we have done? We have changed your this DNA, right? We have changed this gene which was responsible for your blue color of your eye, and this change in the DNA, this has changed the blue color of your eyes, right? So this is basically genetic engineering. That is, if we are changing your DNA or RNA, so definitely the character, that is phenotype, is also changing. We are making the changes in your DNA and RNA, and as a result of this change, what is changing? It is altering its phenotype. So this is there, that is, if you are making changes in your DNA and RNA, it is altering your phenotype. Okay. Now, second is second branch is bioprocess engineering. This is the second branch of biotechnology, bioprocess engineering. What do you mean by bioprocess engineering? This is also second branch. Bioprocess. Bioprocess means some kind of process is taking place. There it was related to genetic makeup and engineering was there. Both the branches are engineering only, right? In one it is related to genetic, and in second it is related to processing, right? So in this, processing is there, some kind of biology, and it is related to processing. Now, what is this branch? In this branch, basically maintenance of contamination free ambience in chemical engineering process for manufacture of products such as antibiotic, with vaccines, etc. is done. Now, this branch you have to understand from the basic. Like, I'll make you understand. Like from DNA, this is the basic process. DNA you must have studied this. From DNA, the process of conversion of DNA to mRNA, it is known as, basically it is known as transcription. Okay, so transcription is the process when DNA it is converted to mRNA, and mRNA it is converted to protein. It is known as translation. Okay, so this is the basic molecular process. This process is occurring in each and every type of cell, that is DNA. So ultimately, when we are making this, you can understand, if we are making changes in the DNA, so what is altering? Definitely, if we are making changes in the DNA, these proteins will be altered, right? In case of genetic engineering also, if we are altering this DNA, if we are making changes in the DNA, then what will happen? Definitely these proteins will be altered, right? So how these proteins are produced? Now inside the cell, these DNA, we are manipulating this DNA, right? We are extracting this DNA, and we are inserting the foreign DNA, and after some time we'll see that proteins are formed, right? So these proteins we can see after some time when we have inserted the foreign DNA, right? So the new proteins, definitely new proteins are different from the original proteins. Why these are different? Because we have made some changes in this DNA, right? So what is this bioprocess engineering? Bioprocess engineering, basically it deals with microbes. And when these microbes are cultured in media, right? So this work is related with basically in lab condition. In lab condition, what we are doing, in lab condition, in labs, you know, this is the petri dish, and we know that we also need certain kind of food. So definitely in microbiology, what is microbiology? Microbiology, basically it is a branch which deals with various kinds of microbes. What are microbes? Microbes are those microorganisms which we cannot see with the naked eye. Like these are bacteria, these are viruses, these are fungi. Now if we want to grow these microbes, how we can grow these microbes? We can grow these microbes with in certain kind of food. We also, if we want to live, how we live? We also require certain kind of food. We require water, we require vitamins, we also need certain kind of food to live, right? Definitely if we want to grow these microbes in the lab, these microbes also require certain kind of food, and that food is known as media. So these are the petri dish, and in this petri dish media is there. So in this media we can grow these microbes, right? So after we can grow these microbes, and after certain period of time we can see that we can isolate these antibiotics, we can isolate these proteins from these microbes in the lab. If we want to isolate these proteins, antibiotics in small quantity, we can grow it in labs. But if we want to isolate these proteins and antibiotics, vitamins and various kinds of products in large amount, we cannot use it in lab. We have to definitely go on higher scale, and definitely we have to go for fermenters and in factories, right? So in labs, so this is basically, this branch is known as bioprocess engineering, right? So when we are using these microbes for the production of antibiotics, vaccines, proteins, what we have to do? We have to culture these microbes in contamination free environment. That means no contamination should be there, right? Because we know that in this air also various kinds of microbes are there, right? So if we will grow any kind of microbe in contamination, so definitely if contamination is there, the product will be contaminated, right? We will not get a good product. Contamination will be there. So bioprocess engineering means maintenance of contamination free. We have to definitely maintain contamination free environment, right? For the products like antibiotics, vaccine, etc. So this is known as bioprocess engineering. So these are the two branches of biotechnology. And genetic engineering, this is basically main branch of biotechnology, and this genetic engineering is also known as recombinant DNA technology. This is the main branch actually. In your board exams also sometimes they ask this question, okay, what is the main application or what is the main branch of biotechnology? So genetic engineering is the main branch of biotechnology, and it is known as recombinant DNA technology. Why it is known as recombinant DNA technology? Genetic engineering, why it is known as recombinant DNA technology? It is known as recombinant DNA technology because we are recombining, we are recombining this DNA, okay? We are bringing some foreign DNA and we are taking this original DNA, and then what we are doing? We are recombining this DNA, and then we are using this DNA for various purposes. So, and we are using technology also. So this genetic engineering is known as recombinant DNA technology, and this is very famous branch, okay, and this is the main application of this biotechnology. Okay, so genetic engineering, I have already told you that this genetic engineering is known as it is also known as RDT, recombinant DNA technology. This is very important, okay? This is another name of genetic engineering. Why it is known as recombinant DNA technology? Because we are recombining this DNA, and this genetic engineering, it is having various applications. Okay, it is used in various fields, like it is used in medicine, it is used in agriculture, okay, it is used in diagnosis. It has vast application, it has various applications. Now what is this genetic engineering? Now the question arises, how we can do this genetic engineering? How this genetic engineering is done? This is very tough, right? Genetic engineering. Engineering, you don't know, engineering itself is very tough. And when we are dealing with the genetic thing, that is, what is the genetic material? I have already told you what is the genetic material. Genetic material is DNA, right? So when we are dealing with this DNA, right? Who has, okay, can anyone tell what, which scientist has given the double structure of DNA? Watson and Crick. Watson and Crick gave the double structure of DNA, right? So genetic engineering, how we can do this genetic engineering? This genetic engineering, the various steps in genetic engineering. Like first and the foremost step is we have to create the recombinant DNA, right? First is we have to create the recombinant DNA. How we can create this recombinant DNA? Now we have to bring the foreign DNA, which we have to insert into the host organism, and with the help of engineering we have to recombine this DNA, and we have to make a mixture. We have to make recombinant DNA, right? Now after that, what we can do? We have to do gene cloning. What do you mean by, what is gene cloning? Can anyone tell what is gene cloning? Gene cloning is, what is cloning? Cloning word means when we are making copy, exact copy, right? Cloning means when the exact copy is made, right? Copying of a particular organism, right? So gene cloning means when we are making copy of particular organism. So when we are making gene cloning, we want to make the exact copy of organism, right? When we have made particular DNA sequence, right? Now we have to clone it. We are making gene clone, right? So we have to make the clone of that particular organism. This is known as gene cloning. Now what is the last step? We have made gene cloning also. We have made recombinant DNA also. Now what is the last step? Last step is gene transfer into the host organism. This is the last step. Now one thing you have to understand. if you want to go from one place to another, how you can go? Definitely you will require certain kind of vehicle to travel from one place to another. So this is the case. You'll go by rickshaw, or you'll go by scooter, or you'll go walking also, right? You can go by any means, right? So if we want to transfer this recombinant DNA into the host organism, we have prepared this product, right? We have prepared this recombinant DNA. Now we can name this recombinant DNA like A, okay? And this is the host organism. This is the human body. Now we have to transfer this A into the host organism. How we can transfer this A into the host organism? Is it possible to directly transfer this A into the host organism? No, we cannot directly transfer this A into the host organism. Definitely we will require certain kind of vehicle to transfer this A into the host organism, right? So this vehicle which will transfer this A into the host organism, it is known as vector. Okay, so this vector will transfer this recombinant DNA into the host organism. With the help of this vector, we can transfer this recombinant DNA into the host organism. Because we cannot directly transfer this DNA into the host organism, it is not possible to directly transfer this host DNA into the organism. It will not work, right? It will not replicate, and it cannot work like this. So definitely it will need a vehicle. Like I can give you the example. If you want to travel from one place to another, you cannot fly and go from that place to another place. You will travel by rickshaw, scooter, or you will go walking, right? You cannot directly fly and go. So this is the case with this only also, right? You cannot transfer this recombinant DNA into the host organism directly. Definitely you will need a vector, right? So vector, what is this vector? Vector is nothing but it is also a piece of DNA in which we will insert this recombinant DNA, and then we can transfer it into the host organism. We'll study this in detail after some time, okay? So third step is basically gene transfer into the host organism. So this is the last step, how we will transfer this gene ultimately into the host organism. Now after transferring of this gene into the host organism, what will happen? Once we have transferred this gene into the host organism, what will happen? Once we have transferred this gene into the host organism, that means we have transferred this A into the host organism. This is host organism, right? Now this is what is this recombinant DNA? This is basically DNA, right? And this host organism is also having, what is this host organism? This must be human also. This must be human being, right? E. coli, any. We must be dealing with animals, we can deal with humans, right? So this host organism is also having its own DNA. So what will happen? What is the purpose of transferring this recombinant DNA? What will happen? This DNA will integrate into the host DNA and it will start replicating, right? So what is our purpose? This recombinant DNA will integrate in this DNA and it will start replicating and start multiplying. And after some time we will see that the recombinant DNA will show its effect, because we have made changes in the recombinant DNA, right? I have already told you, because earlier we were seeing it was having somebody was having blue color of eyes, right? And after altering the gene, we can see that person was having black color of eyes, right? So in this case also, when we have made changes in the DNA, so after some time we can see there are certain changes in the phenotype, right? So what will happen? The foreign DNA will integrate into the host DNA, right? And definitely this foreign DNA will get replicated and it will form multiple copies, right? And this process is known as cloning. That is when multiple identical copies. What is this cloning? Cloning is a process where only identical. Okay, so cloning, what is this cloning? Cloning is a process in which identical means exact copy is formed. Okay, no difference is there. Exact, like photocopy. Okay, these are exact photocopy, right? So cloning is a process in which exact photocopy is formed. Okay, so after some time we can see that this cloning is there in which multiple copies are formed. But all the copies, if 6 copies are formed, then we can see all the 6 copies are exact. They are identical to each other, right? No difference is there. So this is known as cloning, right? Now next is construction of recombinant DNA. How we can construct this recombinant DNA? There are various steps to construct this recombinant DNA. How we can construct this recombinant DNA? There are various steps how we can reconstruct this DNA, recombinant DNA. Now I have already told you plasmids. I have already told you what are vectors basically. Vectors, I have already told you, they are vehicles to transfer foreign DNA host organism. Okay, so vectors are basically, they are vehicles which are used to transfer this recombinant DNA into the host organism. So basically there are two types of vectors, these plasmids and bacteriophages. Two types of vectors are used, one is plasmids and second is bacteriophages. So these plasmids, they are autonomously replicating circular extra chromosomal DNA. Okay, so basically they are extra chromosomal DNA. And these plasmids DNA, they act as vectors. I have already told you these plasmids. What are these plasmids? They are vectors. Okay. And what is the purpose of vector? They are used to transfer the piece of DNA, okay, into the host organism. This piece of DNA, it gets attached to the plasmid. Okay. This piece of DNA, it gets attached to the plasmid. It gets attached to the vector, and then this vector, it transfers this DNA into the host organism, right? So this DNA, it also contains the genes. Now this plasmid, we know that these vectors, they are responsible for transferring this DNA into the host organism. Now there must be certain special properties in these vectors, because these are transferring the DNA into the host organism. So definitely certain properties should be there, certain qualities should be there, which are responsible for transferring these DNA into the host organism. So what are that special properties? They contain certain genes. These plasmids, they contain certain genes which provide them antibiotic resistance to bacteria. So what are these plasmids? They have certain qualities, certain properties, special properties of plasmids are, certain special properties of vectors are there, and you have to study vectors in detail. Vectors are very important. you have to study different properties. Okay, just study the diagram and property of vector. You have to study the diagram and property of vector. What are the different properties of vector and diagram of vector? So there are different properties of vector and diagram. This is very important for this year. Okay, so just study the diagram and property of vector. So these vector plasmid DNA, they contain antibiotic resistance to bacteria, and these plasmid DNA, they cut with specific restriction enzyme. Okay, because this is just a quick revision, right? I'm just doing the quick revision of this chapter, so I have not discussed each and every point with you. So we must discuss this. Okay, what are these molecular seizures? This is also important, right? Molecular seizures, one word is there, molecular seizure. In your board exam, this is asked, what is the molecular seizure, right? So there are certain enzymes. Now when we are dealing with this DNA, right? When we are isolating the DNA from a particular organism, how can we isolate DNA from a particular organism? Definitely we have to cut this sequence from a particular organism and we have to add it in another organism. So how can we cut that sequence from a particular organism? We can cut that sequence from a particular organism by using scissors, right? Because you know that DNA, what is DNA? What is DNA? DNA is basically molecule, right? DNA is basically molecule. So because we are dealing with the molecule, so the scissor which is cutting the DNA, it is known as molecular scissors. Okay, so the DNA, the scissor which is cutting the DNA, it is known as molecular scissors. And what are molecular scissors? Molecular scissors are nothing but they are basically enzymes. Okay. So molecular scissors are basically enzymes. They are restriction. This is very important, right? Restriction enzymes are there. Restriction enzymes are known as molecular scissors, and basically two types of restriction enzymes are there. Restriction endonuclease and restriction exonuclease. Now what is the difference between the endo and exo? You should know. Endonuclease means it is cutting the sequence from inside, right? This is the DNA sequence, and if it is cutting the sequence from inside, it is known as endonuclease. Exonuclease means when it is cutting sequence from outside, it is known as exo. Exo means outside, endo means inside. So when it is cutting the sequence from inside, it is known as endonuclease, and when it is cutting the sequence from outside, it is known as exonuclease. So in this biotechnology, we basically deal with two types of enzymes, that is endonuclease and exonuclease. So basically they are used for cutting the DNA at specific points. What is the special characteristic of this molecular scissors? These molecular scissors, they cut DNA at a specific point. This is the special property, right? They cannot cut DNA. Any specific endonuclease, any specific restriction enzyme cannot go and cut DNA like that only. No. Every restriction enzyme has a specific property. Every restriction enzyme has a specific site. Like A restriction enzyme is there, right? So A restriction enzyme will go to a specific site only, and it will cut the restriction enzyme at that site only, right? So a specific enzyme will cut at a specific site only. This is the special property, right? So molecular scissors, sometimes they ask what is a molecular scissor? So molecular scissor refers to restriction enzyme, and they are basically of two types, endonuclease and exonuclease, right? So you should remember this term restriction enzyme. Now this is one thing, okay, for cutting the DNA sequence. This thing you have to remember, for cutting the DNA sequence, what we are using? We are using the restriction enzymes, right? Now, because for cutting the DNA sequence, we are using the restriction enzyme, right? Now this DNA sequence, after cutting, we have to join this DNA sequence into the host organism also. So we cannot just take this DNA sequence and join like diagonally, right? How you will join a particular cardboard or paper with second paper? You have to join with the help of Fevicol or glue or something, right? So we cannot join this DNA sequence with the host DNA. We need something to join with the host DNA, right? In case of biotech also. So you have to remember, for cutting, we are using restriction enzyme, right? And for joining, second enzyme is there, which is known as ligase, right? In biotechnology, different enzymes are there, and each and every enzyme has a specific function, right? No enzyme is such which we can use for all the purposes, right? Each and every enzyme has a specific function. Like one enzyme is specific for a particular thing, one enzyme is used for cutting, one enzyme is used for joining, one enzyme is used for another purpose, right? So for joining we are using ligase, for cutting we are using restriction enzyme, right? So after cutting we have to join this DNA segment also into the host organism. How we will join this? We will join with the help of ligase, right? Now this is the foreign DNA, right? Now this is the host DNA. We have to join this, both the DNA. Now we will form this recombinant. I have already told you, recombinant DNA. Recombinant DNA means we are combining the both DNA. When we are combining properties of one DNA with second DNA, it is known as recombinant. Recombinant DNA. We are combining this one DNA with second DNA, and we are using technology also. So what is this technique? It is known as recombinant DNA technique. Recombinant DNA technology. And what is this DNA form? It is known as recombinant DNA, right? So this is known as recombinant DNA, right? So recombinant DNA is formed, and what is the technique? It is known as recombinant DNA technology, or we can say genetic engineering also. Sometimes they can ask, right? What is another name of recombinant DNA technology, right? So another name of recombinant DNA technology is genetic engineering.